Consider some mindset shifts for flow cytometry. The first one is that numbers are names. There are a number of CD markers that are used to identify different cells, such as CD20, CD3, CD5, lots of numbers. And it takes a bit of intentional practice to adjust, especially for English speakers, because we typically think of numbers as very separate from names. Some other languages might be a little bit different, such as in Hebrew, where letters can represent numbers. However, for English speakers, it takes a bit of time to adjust so that you are more fluent in the numbers that are names. The second shift for flow cytometry is understanding that flow cytometry is a tool, not an answer. It simply helps you see what you're looking for. And so to say, what did the flow say, makes about as much sense as what did the binoculars say. It really depends on where you're looking. Did you find what you were looking for? In a similar sense, saying that flow is positive is pretty imprecise because it doesn't specify what markers you're looking for. And so it'd be more precise to say flow cytometry demonstrates a population that expresses X, Y, and Z. We'll take a short break and admire nature for a second. But while we're here, have you thought about what it takes to weed a garden? Pause the video for a couple seconds and consider. First, you have to know what is planted. To weed a garden, you want to make sure that you don't pull up the things that are supposed to be there. Second, you have to acknowledge the limitations of your task. When you're weeding a garden, you're not paying attention to the air above or the roots below necessarily, or the components of the dirt. You're looking at the plants. Third, you probably want to check that the plants are normal. They're not wilting, they're not moldy, they're not being eaten by different animals and so forth, and that they're all intact. And then finally, oh, yes, so make sure the plants are intact here. And then finally, look for weeds, the things that are not supposed to be there. We can tell a difference in leaves here in the red solid circled area. Those are different than the green dotted circled area. The green dotted circle are the plants that we expect to be there, that we planted there, perhaps beets in this particular picture. And then the red solid line, or red solid circle, are plants that we did not want in our garden. Interpreting flow is actually like weeding a garden. First step is to be aware of what is supposed to be there. Second, acknowledge the limitations of the task. Flow cytometry usually ignores granulocytes and misses large cells. Third, check for the things that to make sure that the cells are normal. So looking for normal markers and then the presence of abnormal markers. And then look for weeds, whatever is not supposed to be there. You check for any unaccounted for populations. This can be expanded into a 7-step process where you first identify the specimen source, then identify the clinical question, list the expected cells for that source, list the expected markers for each cell and noting any overlap, check for normal markers, check for abnormal markers, then check for any unaccounted populations. Using this step-by-step approach, you can analyze flow from any source. Let's apply these steps to the case that we looked at earlier. Step 1, identify the specimen source. This is peripheral blood. 2, identify the clinical question. Question is this lymphocytosis neoplastic? Step 3, we'll list the expected cells for that source. What cells do we expect to see in the peripheral blood? And what cells do we see within the picture? In this picture, we see T cells, and we would expect to see B cells and T cells within the peripheral blood. Step 4, list the expected markers for each cell, noting any overlap. For B cells, we'd expect to see CD45, CD20, CD19, among others. For the T cells, we'd expect to see CD45, CD3, and CD5. Notice that CD45 is expressed by both sets of B cells and T cells. Step 5, check for normal markers. In our charts, we had CD19 to pull out one population and CD3 to pull out the other population. CD19 is normally seen on B cells. CD3 is normally seen on T cells. So check and check. CD19 matches B cells and CD3 matches T cells. Then we check for abnormal markers. We use CD5 as an abnormal marker. In this chart, CD5 is expressed on the CD19 expressing cells, which is abnormal for B cells. However, CD5 is expressed in the CD19 negative cells, and that matches our T cell percentage. CD5 is a normal marker for CD3, for T cells. Finally, check for any unaccounted populations. As we review our initial charts, there are no populations that we have not check marked. And so we have accounted for all the populations. After you go through these 7 steps, then you can give your interpretation. We do have an abnormal population. B cells are aberrantly express CD5. We would suggest correlation with some type of clonality studies. The T cells are unremarkable. They retain normal expression of CD3 and CD5. So flow cytometry saved us time at the microscope, converting multiple IHCs to 2 charts, which we could then interpret